Today I'm joined by Dr. Katie Bottenhorn. She is a neuroscientist who recently graduated with her PhD from Florida International University, and she's currently working as a postdoctoral research fellow at the University of Southern California. Katie, welcome to the Nature and Nurture podcast.
I feel welcome. It's so fun to hear myself introduced like that instead of like, Catherine is a sixth year graduate student.
Yeah,
Ah, very fun.
yeah, perks of the new degree.
Mm-hmm. Yep. Um, my actual diploma was just mailed to my parents' house, which is a little anticlimactic. I got an email from FIU saying, you should have received your degree. And I was like, oh man, not quite.
Yeah. So that would be a good place to talk about how you first. Well, let's backtrack to undergrad. You mentioned you did psychology and chemistry, which is a very interesting combo I haven't heard of
Mm-hmm.
before.
Yeah, so I started undergrad as a chem major, pre-med. Um, I wanted to be a doctor, like kind of generally. Um, and I hate to say it, but I wasn't like one of those people who's like, I want to be a doctor because I want to help people. I want to be
Hmm.
a doctor because medicine is cool, right?
Uh-huh.
Um, and specifically because the brain is cool. And so that kind of led me. I started undergrad at Auburn University with way too many like high school credits. So I had a lot of flexibility in my schedule, and I'm a big old nerd. So I decided if I could get one degree in 4 years, that would be great. But what if I could get 2 degrees in 4 years? And so then I used to play like, I don't know, I think I called it, I had some stupid name for it, like schedule math, I don't know. But basically tried to figure out how many degrees I could get in 4 years because I was on scholarship. So it didn't
Did
matter
that
how
too.
many credits I took. It was just like, you're going to be here for 4 years, it's paid for, max out your ROI, I guess.
Hmm.
Um, and chemistry is my one true love. And psychology, I was interested in high school, so I just decided, I might as well. Um, and it's funny because it kind of flip flopped in the middle somewhere. I was doing chemistry, I was going to go like take the MCAT, be a doctor, do the whole thing. And then I got into a behavioral neuroscience class where I wrote a paper about the hypothalamic pituitary adrenal axis, the HPA axis, which does the like physiology of stress. And I was like, "What? This is very cool." And the professor was super cool and like one of those like tenured for ages kind of professors. I don't even think he did research anymore. He was just like sat around and talked about the brain and science and stuff. Um, and he got me into a neuroimaging lab, and that's when I decided, or it took me like maybe 15 minutes, and I was like, this is the cool thing, this is what I want to do.
Hmm.
So it started off as chemistry was the serious thing I was doing, and psychology was the kind of like fun, let's do something else kind of thing. And
Hmm.
then it flip-flopped, and at Auburn there was no neuroscience major. There were like two neuroscience classes when I was there, so I took them both. and um, and then I decided that neuroimaging was what I wanted to do with my life, I guess.
Cool. So
Yeah.
neuroimaging work is almost closer to like data science or computer science than it is anything like super bio wet labby. So
Hmm.
did you ever consider with your chemistry background doing something that was more wet lab?
Um, Only extremely briefly. So I've been a computer nerd longer than I've been any other kind of nerd.
Hmm.
I tried to teach myself Python when I was 14. It went really poorly. I did not learn Python when I was 14. I'm not that cool. Um, but when I was in college, I did a lot of different extracurricular research type things. And one of them was a summer research fellowship in cancer biology. And it was all wet lab. I was treating head and neck cancers with different like dopamine agonists. And it was so slow. And I did so many things wrong. And when you do something wrong in a wet lab, you can like see it. It's not like an error message on your screen. It's like you broke
Yeah.
this piece of glass, or you like went to go image your plates and nothing happened.
Hmm.
Um, and that one happened to me a lot, which is really embarrassing. But yeah, I just decided that wet lab stuff was too slow for me. And some of the things in neuroimaging take a long time, like pre-processing your data can take a really long time, running complicated models can take a long time, but it feels to me, it just feels more efficient because I can like do a different thing. I can like, you know, I can go read. I'm still like being productive. But for some reason, I think it suited my like interests a little bit better. And when I started doing neuroimaging research, it was all the like computer based, very I like got deep into the weeds of some of the methodologies pretty early, and I just thought it was the coolest thing. Like when I went off to grad school, my like goal was to find somebody that I could work with who would teach me new things and allow me to learn new things in terms of like analytic strategies and methodologies. So yeah,
Hmm.
just kind of lucked into
Cool.
it.
Yeah.
Hmm.
So what did you plan to study in grad school? Both plan to and then actually study, because those sometimes are different.
Oh yeah. Um, in my experience, extremely different. Um, just like in undergrad, I like really thought that I knew that I wanted to be a doctor when I started. I like was wrong. But in a roundabout way in grad school, it's like the same story and the opposite story. So I wanted to go work for somebody where I could, you know, experiment with and develop new like data analysis strategies. And um, I I did that. Um, it didn't feel like it for a while because when I started grad school, um, there was a new project about educational neuroscience going on. And because of that project, my advisor had gotten some funding from some people in the business school who were piloting their new educational video game. And so she asked me before I like arrived at FIU, she was like, "Hey, do you want to work on this project? You can say no, we'll put you on something else, doesn't matter. Um, but it seems pretty cool and it might be a good fit for you." And so I started out studying like video games in the scanner, like how do you do that practically? How do you like have someone play an immersive video game while also treating it like an fMRI task? Um, and how like what changes in like an adult healthy person's brain happen when they're like learning. So I am not interested in educational neuroscience. I like think it's cool in like some corner of my brain that's just like casually interested, but I was really interested in the like, you know, developing new cool tools kind of thing. So
Hmm.
unfortunately, that funding fell through, I think, after a year, and nobody told me. So I just stopped getting paid mysteriously one day.
Man.
Yeah, no, it was great. Um, and my advisor just kind of scrambled and put me on a different project that was geared towards neuroimaging meta-analysis, which I'd done a little bit of in like undergrad and grad school. So I was familiar. It wasn't like my bread and butter, but might as well. And then and then that grant fell through. Oh my gosh, it was an adventure. Um, and so that's the story of how I didn't get paid for a month and a half in grad school. Um,
Darn.
but then I got put on the ABCD project, and so I'm not really a developmental person, but I got to work with, I like working with kids, it turns out. I like working with big
Uh-huh.
data, so that was fun. Um, and oh my, this is a really long answer. Sorry, I didn't expect it to be this long. Um, but for the longest time, I thought that I wanted to develop new cool tools, and I pushed on that, and I got a little bit of pushback from my advisor because she kept saying, no, you need a content domain. You need to find a thing so you can study the thing and be the expert in that thing. And I was like, I don't care. Pick a thing. Like, you can pick. I don't care at all. Because I just wanted to do the new cool tools thing. And then we had this meeting one time, and she started it with, I just want to apologize. And I was like, uh-oh, did something happen that I wasn't aware of? Like, am I not going to get paid again? But then she said, she's like, I think I like gender stereotyped you and told you you couldn't be like a methods person, like a computery code writing analysis, I don't know, designing person because you're a woman. And I was like, I had not considered that, but thank you. Because she is really geared toward, like her research program is geared toward, or at the time was geared toward like addressing the leaky pipeline in science, technology, engineering, and math education for women. So she was like, "Oh,
Mhm.
did I just accidentally make the pipeline leak?"
What do you mean by leaky pipeline?
So the leaky pipeline is an idea that the reason there's less representation of women at higher, progressively higher levels of education and careers is that they just kind of like fall out at various points upon the like pipeline. So that could be anything from like being told in high school that like girls aren't good at math, so you don't like if you don't think you're good at math, then you get to college, you perform poorly because of stereotype threat, and you don't go into math heavy careers. It happens in physics too. Physics is very male dominated, and it's kind of like a self-fulfilling prophecy in some ways. Like the further you get, the more men there are, and so the less women feel like they belong. I think the general idea of like the leaky pipeline is pretty debated these days, because the more people, men, looked into it, the more they realized women aren't just like leaving, they're kind of being pushed out. So leaky
Mm-hmm.
implies passivity, and it's not necessarily leaky. But anyway, so
Mm-hmm.
my advisor in graduate school, Angela Laird, was a physicist, is a physicist. Her like education's in physics. She did her PhD in physics, and she slid from physics into neuroimaging. So it was really close
Mm-hmm.
to her heart, and her experiences like figuring that whole thing out. So after we had that conversation, I got a lot more exposure to the sorts of opportunities that I wanted in terms of like developing professionally as somebody in the like methods development sphere. And I fell in with a bunch of really cool, interesting, like the people themselves are interesting, and like the work they do is also interesting. So like it's more like the work they do is interesting. I'm sure there are interesting people everywhere, right? But I kind of fell into the neuroinformatics sphere, and I really like it there. So
Uh-huh.
in my fourth, third or fourth year of graduate, they all kind of run together. I had just about finished writing my dissertation proposal. I was going to look at the ABCD data set and do some cool stuff. I honestly don't even remember what was in there.
Mm-hmm.
And then another person in my lab who is also into methods development, and software development and all that sort of thing, he proposed an idea for a project that he wanted to do that was focused on like very, very many imaging samples or imaging time points of very few people. Um, so it's like the dense sampling approach or deep phenotyping, or there's like a million different words for this approach. He wanted to do it because you could do some really cool like machine learning and um, I guess like predictive modeling sorts of analyses with that sort of data. But he only really cared
Mm-hmm.
about the first time point and the last two time points out of like 13 or 14.
Mm-hmm.
And so he gave this presentation in lab meeting. He was like, does anybody have any ideas for like what else we could do in the middle? Like he didn't really care as long as something was happening. Um, and I was like sitting on my hands because I have a bad habit of volunteering myself for things that I really shouldn't be doing, that I don't have the time to do. And the next meeting I had with my advisor, she said, so what do you think of Taylor's project? And I was like, well, kind of hate that I'm interested in it, but I'm interested in it. And the funniest part is that at lab meeting, I was being kind of snarky. And the other PI that's like in charge of our lab said, how are you going to balance your sample if it's only 3 participants in terms of gender? And I was being snarky and I said, we're not going to, no boys allowed, only women. And my advisor was like, no, only women. That's what we're going to do. Like, seriously, only women. And that's the story of how I ended up changing my dissertation after it was all but proposed to look at variability in the brain with respect to the menstrual cycle and ovarian hormones, which is a fun callback to my one true love, chemistry. Human biochemistry is my jam, so I was really into getting back into that sort of thing and thinking about cell signaling and how this all happens on a
Yeah.
molecular level. And so just like I said at the very, very early beginning of this answer to your question, I didn't have a content domain until that moment, and I realized that like my content domain, like if I'm going to have one, is women's health and reproductive health. Like
Uh-huh.
that's what I'm really interested in. Um, some like just like being a woman kind of like made me more likely to be interested in that because of like how little information there was about specific questions that I had regarding my own health. That lots and lots of people have, because there are so many women in this world, and women and people who, you know, reproduce, people who menstruate. There's lots of different ways to group people, but I was super interested specifically in menstruation and contraceptives and how that is happening. Um,
Hmm.
so, unfortunately, that really cool project that we spent all that time putting together launched in February 2020. Um,
Right.
so
With
we
the
got
right before the pandemic.
we got maybe a third of the data collected, but I still managed to squeeze in some stuff into my dissertation about women's health and reproductive health. So
Yeah,
go
so
team.
there's a lot to unpack there. So before we
Mm-hmm.
talk about your dissertation, I want to ask more about the method side of things because,
Yeah.
you know, there's some people who will go into research more like hypothesis driven or more idea driven, and then
Mm-hmm.
later they learn the methods. And then there are other people who might come from like a computer science background and then only later learn the research. And it seems like you started off well with psych and chemistry, and then you gradually became more interested in like the big data stuff. So do you think that offered you a different perspective as opposed to someone who might have been doing the same research from with like a CS background?
Yeah, definitely. Um, I like to. I like to joke, or some people, and I like to joke that um, like my code's always going to be kind of garbage because I'm not a computer scientist. It's going to do more or less what I want it to do. It's going to do exactly what I tell it to do, which is unfortunate sometimes. But I would have liked. Sometimes I like think to myself, if I could have redone undergrad, like what would I have done? And I usually end up with probably computer science or math, like a degree in computer science or math instead of chemistry or psychology. But the problem is that if I did that, I wouldn't be where I am now, and I like where I am now. Um, but I was just talking to my boyfriend this weekend. I was like, I should have been a mathematician. Like everything, all the like methodsy cool things that like I'm really interested in, and if I like Google it a term and I'm like, how like if I had learned about this in college, how would I have learned about it in college? Like what class would that have been? What major would that have been? Just to kind of wrap my head around like where to find more information. It's always math, which sucks. I'd love to be better at coding, but in terms of like, I think a lot of the really cool big data techniques and more nuanced ways to understand the brain, especially, but big data in general, are deeply, deeply rooted in some of the higher level mathematical topics that I never would have learned or never had learned. I didn't take any math. I took one math class in college. And it
Mm-hmm.
was for fun because, like I said, big old nerd. It was linear algebra, so great choice because everything I
Hmm.
do is linear algebra. But yeah, so
Hmm.
I like graph theory. Graph theory is math. I like complexity theory, also math. I like clustering algorithms, math.
Hmm.
High dimensional data structures.
Hmm.
So kind of sucks, but
Right.
here we
So
are.
with that methods work, you get to well make new methods that can be used for any type of research, but on the other hand, you don't get to see it. And you mentioned you found this passion of yours in women's health. So you
Mm-hmm.
at least you got to apply your research and start seeing the results to come out of it.
Yeah, and that's kind of that's what my advisor has done with her career that I'm kind of hoping to also do with my career. And I think to an extent, Dr. Herting also did it with her career, being really like interested in and reliant on the methods. And um, my thought was just to apply them to things to show people that like, look, I can do both. I can be, math and science lady and do the computer stuff over here, but I can also like, I do know stuff about neuroscience and neuroimaging and the way the brain works. So it's a balance for sure. But one thing that I really love about all those experiences is that I have the skills to do both, and um, being able to turn novel analytic strategies on big data is just a good thing right now. Like that is a marketable skill.
Mhm.
Um, and I still don't really mind like the idea of being a hired gun. Like that is a skill that I have. Point me at some data and I will use my skill. Tell me the question
Mhm.
you want answered, and here we go. Let's answer the question. But the other cool thing is that as neuroscience is getting bigger, it's also kind of like sliding into like precision medicine and precision neuroimaging and like deep phenotyping.
Hmm.
And people are learning that you can't really understand some of those things and some of the aspects of like brain structure and function without understanding hormone fluctuations. So it all
Uh-huh.
comes full circle.
Right.
You can't understand development without understanding puberty, and you can't understand puberty without hormones. You can't understand brain network variability over time if you don't understand it in men and women, and you can't understand it. I mean, men have circulating hormones too, of course, but there is a great deal of research just starting to delve into like exactly how much hormonal fluctuations change the brain structure and function. So it I don't really have to choose. I can do both. I am doing both. It's pretty great.
Yeah, that is great. So traditionally, neuroimaging research is done with like one scan or maybe a couple scans at a start point and an end point if it's longitudinal. And then bigger or better studies were just seen as getting more people. And what's new is now doing this whole series of scans
Yeah.
to measure like what was the word you used? Like
Dense sampling is one term. That's the term that we
uh-huh.
used. Deep phenotyping is a little bit like cooler sounding,
Uh-huh.
but it means the same thing. I'm sure there are more. They're not. I can't bring them to mind at the moment, but yeah, there's it's a new idea. I don't know if it's a new idea. I'm sure lots of people have been doing, dense sampling type approaches for a while now, but
Hmm.
it is just now becoming more feasible in neuroimaging. And there are a couple of really cool examples that have already shown us super neat things about the brain. One of them is the Midnight Scan Club, which they scanned at midnight, hence the name. They could get a discount on scan time if they scanned at midnight, and so they decided to go for it. I think it's only like 10 or 12 people, but they have like hours and hours of data, imaging data per person. Um, and one of my favorite papers in grad school, that is still pretty favorite, that was the basis of the like end chunk of my dissertation, um, is or looked at, using those data looked at the magnitudes of variability due to like differences between people, differences within a person between tasks, differences within a person between like time points, etc. And it found that the differences between people in terms of like functional connectivity in your brain are pretty significant, um,
Hmm.
and that really really pushed me as far as like methodology development was concerned into looking more and more at individual variability,
Hmm.
um, which
Right.
is
So on
just
one
the
hand,
coolest.
that completely makes sense that people's brains are different. And then on the other hand, every intro neuro class is like, here's the brain and here's all the different parts. And they show it as if like they're always in the same areas.
Mm-hmm. And anybody who's ever tried to segment a brain knows that that is not the case. Oh goodness gracious me! But yeah, it's cool. I feel like that's the transition from undergrad to grad school for regardless of like what field you're in or where you study is there's a little bit of unlearning that has to happen.
It's like everything
Um,
you know is wrong.
mm-hmm, mm-hmm. I mean, it happened in calculus when you like learn how to do integration by parts, and then or not integration by parts, when you learn how to do like I don't know limits or something, and then you move right over to doing derivatives, and you're like, oh my god, this is so much easier. Are you kidding me? We could have been doing this the whole time. Um, it's not easier to learn that the brain is so very different between people, but it is a similar sort of paradigm shift. You're like, oh my god, it's okay. Thank you for teaching me the wrong thing. Or the less useful information, I guess. It's not technically wrong, but it's not so useful.
Uh-huh. So is the high interperson variability is the reproducibility crisis that's happening in some areas of science, is that also impacting neuroscience for this reason?
Oh, for sure. Definitely impacting neuroimaging. I don't know if it's. I think okay. Yes. There's a problem with neuroimaging that has been, I mean, slightly addressed, but it's very difficult to address because neuroimaging is expensive. Scan hours are like $500 roughly. More some places, less some places. And so a lot of early studies kind of stopped at like 15 people, 20 people, 30 people. And just given the amount, like enormous amount of complexity in the brain, and the enormous amount of noise in functional neuroimaging data, it turns out to have a sufficiently powered study, you need the orders of magnitude more data than that. And one of the fun slides I had in my job talk, dissertation defense, and the presentation I'm giving on Wednesday, it's based on a simulation study that looked at different, with different, let's see, like effect sizes and different sample sizes. What's the probability that your significant effects overestimate the effect size? And what's the probability that you actually have the power to detect your effect? And with 20 people and almost every effect size, you are 100% overestimating your effect size with your like significant findings. And so that's that's a pretty big issue for reproducibility.
So it goes down with more people, right? And you mentioned you're doing work on ABCD, which is this national adolescent brain and cognitive development study with like, what is it, 13,000 kids over many years?
Yeah, it's the goal, I think, was 11,500 over 10 years. I think they overshot it for the baseline because retention is just an adventure, especially over 10 years, especially for a neuroimaging study. So, yeah, it's great. There's a kind of idea in neuroimaging that there are two ways to solve this problem, or to address this problem, not to solve it. The first is more people. The second is more data per person. And there are trade-offs, just like in literally everything in life. But so the ABCD approach is super cool because it's population, or it's approaching population level. More people, more data, longitudinal, so many measures, imaging and not imaging. And then dense sampling and deep phenotyping is like the other approach. It's like fewer people, much more data per person. And so some of the stuff I did in grad school looked at, how much variability is there in a person from day to day, and like how much does that matter depending on what you're studying. And the ABCD approach is more like how much variability is there across the population, and what does that matter for understanding, the consequences of different early life experiences and environmental impacts and sociocultural impacts, et cetera, for eventual drug and alcohol use. And so it's just a really cool way to think about neuroimaging right now. Is like there's, we agree that there is a problem, there's obvious gaps in the literature, and there are lots of good ways to fill those gaps. And so, yeah, I can't decide which one I think is more interesting.
Right. So going back to your dissertation research, you're looking at hormonal fluctuations in women and how that relates to brain changes. And well, this is over a relatively short time span, right?
Yeah.
So I wouldn't expect there to be much change, but I'm guessing you're gonna blow me away and blow our audience away with like, here's how hormones are changing your brain in ways you'd never expect. That'll be your BuzzFeed title.
Sure. Somebody else already wrote that paper, and it was really cool. And I borrowed their data because, like I said, my dissertation project got the axe. So I was more interested in like relative magnitudes of variability. Specifically, like I was looking at hormones and stress and sleep and hormonal contraceptive use. And so because things got cut short, I couldn't do all the beautiful, wonderful things that I wanted to do with the like crazy statistical models I wanted to dream up. But instead, I decided it would be interesting to know just how much things change. So there's a really cool paper published maybe a couple of years ago now about changes in brain structure over the course of the menstrual cycle that found that there is just a wild change in the size of your hippocampus over the course of the menstrual cycle, like wild. And so obviously that has consequences for, people who menstruate it also has consequences for anybody who studies learning and memory, because if presumably, ideally, half of your sample has these wild fluctuations in hippocampal like structure happening, um, and you, for instance, maybe haven't asked any questions about the menstrual cycle, or haven't like tried to control for that or account for that, even or like even know about that. You can't really do a good study of learning and memory without taking those sorts of things into account because of the massive confound that is the menstrual cycle. So my research was significantly less groundbreaking than that, but I found that some of the like places in the brain, some of the brain regions that vary the most with respect to the hormones that fluctuate over the course of the menstrual cycle are also highly variable with different levels of stress and sleep. And so it's not super duper cool, but it does have the same sorts of implications, right? Like if you're going to
Hmm.
study stress and emotion regulation and sleep and all sorts of things, your effects could really be washed out by menstruation if you're not going to account for it.
Hmm. Right. So when we're talking about wild differences, is that like, maybe a millimeter change, but from a scientific standpoint, that's huge? Or is it like if we could hypothetically look at your brain in real time, would we see it like sort of ballooning? And
You know, that's a great question. I don't actually know the numbers on that one. It is small in terms of like actual size, but I think in terms of like the proportion of like the total size to the change or something like that, or the like how related it is to progesterone levels specifically, I think it's relatively high. Um, but yeah, I mean, it's one of those science things. It's not like your hippocampus takes over your brain in the luteal phase of your menstrual cycle. That'd be wild, but
right. So then, do you know of any research linking that to behavioral changes? Because you could either make a case for brain changes and then the brain changes change behavior, or you could say maybe something like your behavior changes and then your brain sort of maps onto that.
yeah. So that's kind of an interesting thing about neuroimaging and psychology research is causality is difficult, if not impossible, to determine, just because we have ethics when we study human beings, and there's only so much experimental manipulation you can do, right? But that research, like I said, is relatively recent, last couple of years. Before that, there's a lot of research looking at changes in like working memory, episodic memory, different, skills and cognitive domains over the course of the menstrual cycle that finds like interesting things. Like I think um, there are some changes in verbal working memory. I don't know if that's true. But I remember something about there being changes in memory and word processing with respect to the menstrual cycle. And so, memory changes, hippocampus, is a logical leap. But in terms of causality, the fun thing about studying um, biochemistry in the brain, especially when it comes down to the menstrual cycle, is that the hormones that fluctuate over the course of the menstrual cycle do have central nervous system actions. Um, and so these are hormones that are signaling in the brain, and in the case of progesterone, hormones that have metabolites that are major neurotransmitters. Um, or agonists of major neurotransmitter receptors. So it's cool to think about in that way. I'm not sure that you could really make the case that behavior is driving these changes, just because behavior is so bananas, right?
Hmm.
Like I don't know that anything that I do like habitually is on as regular a cycle as like my menstrual cycle, so that would be
Hmm.
a little bit tougher. Um, but that's kind of one of the things that I like so much about um, biochemistry, human biochemistry, the brain, and how they all interact is it's kind of a dumpster fire. Like everything is touching everything else a little bit here and there. Um,
Hmm.
Hormones have like direct signaling in some cases, and their metabolites have like agonistic or direct signaling in other cases. So teasing these things apart is really difficult,
Hmm.
but it's super cool. Um, so that's one of the reasons I want to learn about things like complexity theory and chaos theory, is because like it's a disaster, it's chaotic. Like, give me some fancy math to help me make sense of
Uh-huh.
it all, right?
Right. So it's like a small hormonal change, or really any initial change can have like a huge butterfly ripple effect type
Yep.
thing.
Mm-hmm. definitely.
So if you see these changes just in adults, I imagine that over the course of development, the effects would be uh, even larger as everything's just changing and hormones are going wild. So
Yeah, especially because your brain isn't done cooking yet.
yeah,
That's part of development, right?
uh-huh.
Yeah, I wish I knew more about puberty. Maybe I'll have to learn about like how hormones change over the course of puberty.
Hmm.
Um, but yeah, I can't even imagine. I mean, I think I like remember being a teenager fairly well. I remember it being very confusing and difficult. I think lots of people feel that way. I think people who like are parents of teenagers would agree. Um, and so I can only imagine that like some of that difficulty is just because there's a mismatch in like I don't know, like how your brain is going, how your hormones are going, how you feel as a person. Um, I used to joke that I had never felt more like an adult than when I was like 17-ish, 18-ish, like figuring out college things. I was like, I should make my own decisions and do my own things, and
Hmm.
um, that's pretty late in development. But you're not done until you're like in your 20s. Um,
Right.
so yeah, it's just it's wild. So you on the one hand, like you're becoming more autonomous and more in charge of your own life, um, but like you're not quite ready to be all of that and to do all of that. So and then like I don't know, the more I learn about social development, especially and like high school, the more high school kind of makes sense in a weird
Hmm.
like abstract way. Like we're all just trying to figure out how this whole thing works. It's kind of
Hmm.
messy.
Yeah. So what are your plans for your future research during this postdoc?
That's a great question. So my first project is doing. It's a little bit related to my grad work. I'm trying to parse out variability in the ABCD dataset, specifically in the brain changes over the first 2 years. So imaging visits are every 2 years. So the project was started, I think data collection was started in 2016. So we had imaging visits at baseline, so 2016 to 2018, and then kiddos started coming in for their 2 year follow ups. So we have 2 time points on most everybody. And then in 2020, when their 3 year follow ups was started, things got a little out of hand. So it's cool that we have thousands and thousands of like 2 time points of individuals and their brain function, their brain structure, all sorts of things. And there are a lot of developmental studies with smaller samples. And there are a lot of developmental studies that have looked at variability and change. But so far, looking at brain change and the variability and the heterogeneity in brain change on this level hasn't really been done. Um, so I'm going to start looking back at variability between people, um, and and just how much there is going on in terms of like you know
Hmm.
mid childhood development. That's not early childhood because
Uh-huh.
that's much younger people, but yeah. So I'm gonna get to turn some of my favorite like big data techniques on this big data and see if we can't make sense of that. But
It's
overall, I want to get a little bit more experience with the big data. I want to start looking more into um, like puberty and hormonal change
hmm
in that time, but that'll be a little bit further down the road, I think. And other than that, I'm just excited to learn about some of this stuff.
hmm
I'm going to eventually study the effects of air pollution on brain development. And that'll be super cool because I don't know anything
hmm
about brain development, but if you blow up my interest in like women's health, reproductive health, the menstrual cycle, and how that is related to variability within a person, if you kind of like explode it a little
hmm
bit, um, I'm just interested in how much and why the brain changes in like
hmm
regular people just living their life. And I think air pollution and environmental health plays into that a big bit. So
Yeah,
I
that's
get
very
to,
exciting. So first, the goal is to see how much variability there is between kids and their brains, and then only once you establish that, you're going to look at all these additional things like air pollution and hormones and stuff like that.
Yeah, well, I don't know exactly how like linear that process will be, right?
Right.
I would like my investigation of the heterogeneity in brain changes to have like a capital R result, like we found there are different groups of
Hmm.
people developing differently, and then that would be great to like branch out from there and look at
Hmm.
like are these differences cognitively relevant? Are they related to your environment? Are they related to puberty, which I think would be really cool.
Hmm.
There's some there's some literature that says that there is much more variability in the brain like after puberty starts than before, which is really
Hmm.
cool. I don't
Yeah.
know a lot about the developmental literature, but that would be a really cool thing to dig into. So I'm hoping that it proceeds like that, that I can just like, look, I found a thing, and then self-cite in another
Uh-huh.
paper. But I've had a lot of really interesting research adventures over the past few years, and it doesn't always go quite that well. So fingers crossed.
Yeah, fingers crossed. That sounds exciting.
Mm-hmm.
Thank you very much for your time, Katie.
Yeah, I'm always happy to chat about science.
Same here.